for the second part of the video regarding the cells, so we'll be discussing about the eukaryotic cells. So the eukaryotic cells are cells that contains membranes, or rather endomembrane system. You have organelles inside. So their DNA, the most unique or central classification or feature of eukaryotic cell is that their DNA is contained inside a nucleus. So they also contain other membrane-enclosed organelles. So those membranes allow the organelles of the eukaryotic cells to regulate the substances entering and exiting, and they provide specialized environment for its specific function. Basically, they compartmentalize their functions. So for example, one organelle is involved with breaking down of substances. So the interior of that can be very acidic or very basic depending on the need. So that is separated from the rest of the cell so that it will not affect other proteins or other materials inside the cell. So we have several components of the eukaryotic cell. So these are the membrane-bound organelles. So these are organelles of the cells that have membranes. So the first one is the nucleus. So the nucleus protects and controls the access to the DNA. The endoplasmic reticulum is the one responsible for the modification and the site of the synthesis of polypeptides and lipids, among other tasks. And then we have the Golgi Body, which is for the modification and packaging of polypeptides and lipids. We have the vesicles. It transports your polypeptide products, or even it transports harmful materials. It can also serve as storage, or it separates the harmful substances from the rest of the cell for breakdown. And then we have the mitochondrion, the powerhouse of the cell. So it's the one responsible for the production of ATP, which is the energy currency of your cell. And then you have chloroplast for plants. So you have your chlorophyll there. So it's the one responsible for making sugars, glucose. So lysosomes is the organelle responsible for the digestion of harmful substances, as well as the breakdown of garbage or waste materials. So we also have peroxisome, similar to lysosome. It's for the breakdown of fatty acids, amino acids, and toxins. The difference between both of them is how they actually break down those materials. So the other one is vacuole. It's for the storage of food or waste. So we also have organelles without membranes. So these are the ribosomes. Ribosomes can also be found in prokaryotic cells. So these are the ones responsible for the synthesis of protein. Basically, they are the ones that are synthesizing proteins, and they are found in the ER, or your endoplasmic reticulum. That is why the ER is your site of protein synthesis. Now we also have the centriole, which is which anchors the cytoskeleton. Its function is during mitosis, or the cell reproduction. And other components, these are the cytoskeleton, which is basically the one that gives shape to the cell. It also provides the cell means of locomotion, as well as it helps in transport vesicles. So these are electron micrograph images of the cell. So in here you have your animal cell. So this is a white blood cell of a guinea pig. So the large structure here, this one is actually your nucleus. So a large component of your eukaryotic cells, some eukaryotic cells, a large part of them is made up of the nucleus. And then this one here is your endoplasmic reticulum. And then you also have smaller organelles there. So these are your mitochondria. So it's considered sausage shape. So if you look at it closely, well, it's not exactly sausage-like, but you have creases here. So just like a parang gatla-gatla. So you have the mitochondria. And then this is for the plant cells. The plant cells, you have a prominent structure for that. They have cell wall. So this is your cell wall. So in here is your nucleus, and then this is your vacuole, central vacuole, storage of water, materials, and others. So this is another vacuole. Okay. So that is an image of your eukaryotic cell. So if we discussing one by one each component, so we have the nucleus, which contains the DNA of the cell. It separates the DNA from the rest of the cytoplasm. Effectively, you are protecting the integrity of the DNA so that you do not mutate, the genetic material is not easily destroyed or mutated. So inside the nucleus, there are other components or other structures. You have the chromatin, which is basically the DNA with the associated or support proteins in there. And then you have the nucleoplasm, the semi-fluid interior of your nucleus. So it's like a mini cell inside the nucleus. So the nucleoplasm is like the cytoplasm. And then you have the nuclear envelope, a double membrane. So double membrane, that means 2 layers of membranes. Double membrane system with nuclear pores. So it's pockmarked. It has many holes in there so that the mRNA can easily pass through. And also you have the nucleolus, a dense region of proteins and nucleic acid where ribosomal subunits are being produced. So here is a structure of your, an artist sketch actually of your nucleus. So the nuclear envelope is a double membrane system, and then these pimple-like markings, these are basically the openings, or these are the holes in your nuclear envelope. So that is where your mRNA passes through towards the ER. So the ER is basically integrated, almost integrated with the outer layer of the nuclear envelope. So it's almost fused. So that when your mRNA passes through, so immediately it can go to the ER for the information to be translated to proteins. So you have the nucleolus, a dense region of your proteins where the ribosomal RNA are being produced, and then you have your chromatin, so inside the nucleus itself. So the structure of the nuclear envelope, so as you can see, it has many pores. So the pimple-like structures, these are the pores. It's actually a channel where your mRNA can pass through. So this is the actual electron micrograph image of your nuclear pores. So they have basket-like structures on the interior. So this is the basket-like structures. So this is another image showing the cross-section. So it's actually a passageway. The nuclear pore is a passageway to the nuclear envelope. Okay, so the endomembrane system is actually the series of interacting organelles between the nucleus and the plasma membrane. So the components of that, you have the endoplasmic reticulum, both endoplasmic reticulums, the rough ER and the smooth ER, the Golgi body, and the different vesicles. So they function in the making, modification, and transport of materials, which are proteins, usually proteins and lipids, for secretion or insertion into the cell membrane. They are also responsible for the detoxification of cells, recycling of wastes, and other things. So part of the endomembrane system is the endoplasmic reticulum. We have two, rough ER and smooth ER. It's called the rough ER because when you look at it in the microscope, you have many granules inside. It's like very sandy structure. The reason for those granules are actually the ribosomes. So the rough ER is the one with the ribosome. It's the site of protein synthesis, and it's responsible for folding polypeptides in their tertiary form. So it's very important in creating proteins and folding them into their proper structures. And then we have the smooth ER. Smooth ER because there's no ribosomes there, so they have no granules. So this one is responsible for the synthesis of the lipids, breaking down of carbohydrates and lipids, and detoxification. And then we have vesicles. Vesicles are small membrane enclosed sac-like organelles for storage and transport of substances. And then we have peroxisomes, vesicles containing enzymes that break down the hydrogen peroxide, alcohols, and other toxins in the cell. And then the liposomes, this one now contains enzymes. So this one, both of them actually contains enzymes. So depending on the type of enzyme they contain, so you have the lysosomes and the peroxisomes. So these lysosomes are vesicles containing enzymes. They fuse with vacuoles to digest waste materials. And then the vacuoles are vesicles with various functions depending on the cell type. So, for plant cells, the vacuoles are storage of water, mainly water. For animal cells, the vacuoles is more of storage of other materials like, for example, PHB, polyhydroxybutyrate. For PHB synthesizing cells, these are the storage for the polyhydroxybutyrate. And then you have central vacuoles, occupying 50 to 90% of the cell interior. This is mostly found in plants. They store amino acids, sugars, ions, and bases, and also regulates fluid pressure in plant cells. So another part of them, endomembrane system, is the Golgi Body. The Golgi Body is a folded membrane that contains enzymes. They are the one that are responsible for the modification of proteins and then as well as packaging. So they package finished products to the vesicles and carry them to the plasma membrane or to the lysozymes. So this is how the system, the transport system inside your cell functions. So you can see vesicles going in and out. So for example, you have products being synthesized in the ER, and then you have your these products are transported via the vesicles. So you have the ER here, then transported via the vesicles to the Golgi Body. So depending on the fate of that product, so they can be used, they can be transported to the outside surface of your cell for release, or they can be fused with lysozymes for degradation. So depending on the fate of your the contents of that cell. So that's it for now. So in the next part of the video, we will be discussing about mitochondria, chloroplast, and the cytoskeleton.